Hi, this is Ziyun from Carleton College. I'm presenting on our research about a weighted distance-based approach for deriving consensus tumor evolutionary trees. I want to thank ISMB for this opportunity. For the structure of my presentation, I will introduce tumor evolution and tree structure first. I will then introduce the consensus tree problem that our research focuses on, and of course, our method using integer linear programming and the testing on real data set. As background information, and as most of you might already know, cancer is due to the development of a tumor. The healthy cell gains somatic mutations, which allow it to replicate without control. Descendants of the cell might require new somatic mutations. Some of these mutations will also provide a selective advantage, leading to a new increasing population. As a result, at any given time, a tumor is a heterogeneous mix of different cell population, each with different mutations. However, note that we don't directly observe the evolution of a tumor. Instead, we only get a glimpse of the current state of the tumor by sequencing tumor DNA sample at the time of diagnosis. From this data, we want to infer the evolutionary history of a tumor. Specifically, our group represents the evolutionary history as a rooted tree. The vertices represent cell populations, and the edges represent direct ancestral relationships. For example, A.22b means the cell gained mutation A first, and this population of cell gained mutation B subsequently. This is what we want to infer. Inferring the evolutionary history of tumor has very important implications on how we treat cancer. For example, if we want an effective treatment plan for the patients, we want to target all cells in the tumor, both the blue and yellow lineages, in order to cover all lineages in the tumor and may be more effective together when considering treatment plans. There are many different algorithms out there that have been developed in the last decade to infer such clonal trees. This method can be designed for different data types, from bulk to single cell, and they all have different algorithm approaches and assumptions. These different methods have different strengths and don't always agree. We need a way to better utilize this broad spectrum of method, especially when these multiple data types are available for individual patients. This leads to the consensus problem we've been working on. As different methods produce different plausible tree for one single tumor sample, if I am a doctor, I don't want this many trees, and I want one that best summarizes all of these possible trees. A consensus of all of the trees is a more accurate representation of the tumor's true evolutionary histories, and this problem is what we've been working on. There are several existing methods that's been working on this problem, and they have certain limitations. For example, previous method by our groups is called graphic. It's a graph-based method that aims to find a tree that minimizes the total distance from the consensus tree to all the input using a simple distance measure called parent-child distance, which only captures relationships between parent and child mutation pairs. There's also country DP, which took a different track and wasn't distance-based. Instead, it aimed to maximize the number of directed partitions in consensus tree, but they all still treat all input trees equally. In our work, we introduced a method called TULIP that addresses all of these limitations. It stands for Tumor Evolution Integer Linear Programming. Let's introduce our method we formulated the problem as weighted M tumor tree consensus problem. The input include a set of plausible tree with their corresponding confidence weights. These weights all sum up to 1, and higher weights means higher emphasis should be given in the consensus tree. We also have a distance measure. Specifically, in our research, we used ancestor-descendant distance, which means that if A is pointing to B, then A is ancestral to B, and also A is ancestral to B's descendant C, and also D. And this distance is calculated by the absolute differences in number of ancestor-descendant pairs between the trees. For example, the only difference here is from B to D is not represented in the second tree. So the distance is 1. For the output we're trying to find, we aim to minimize the weighted sum, and we're trying to find a medium tree that minimize the weighted distance to each input trees. The approach we used consists of 2 steps. First is an ILP integer linear programming step, which is a computational technique for solving optimization problems. Many different problems can be formulated as an ILP. We formulated our problem as an interesting one and implement in Python to use the solver to solve it. So we used the input trees and weight to identify a directed acyclic graph that contains all the ancestral relationships in the optimal tree as the first step. And we then perform a transitive reduction on that graph to get to the tree by removing the redundant edges that already been represented by the main branches of the tree. Now, looking at the details of ILP, there's an objective function which we define to minimize the weighted total distance from consensus to all the output. There's also variables we introduced to represent a tumor tree and some linear constraint to help restrict our output to a valid tree structure. First, let's look at how we define the variables. The x variable we used is to indicate edges. So if xab is 1, that means A is ancestral to B. The R variable is used to indicate whether the mutation is root or not. RA equals 1 means A is in the root. And in this example, all other mutations are not root, so their all variable are all set to 0. There are constraints set for this variable to make sure it represents a valid tree. First, we ensure there's at least one root by these equations. And also, we use these 2 constraints to make sure root is ancestral to all other mutations. So the output is a connected graph. Here in the first inequality, we require that all mutations labeling the root are ancestral to all other n-1 mutations. And similarly, since I only want one root, any mutation that doesn't label the root must be ancestral to fewer than n-1 mutations coded by the second inequality here. We also need to ensure that ancestral relations output by our method are transitive, which means that if mutation A is ancestral to mutation B and B is ancestral to C, then we need to infer that mutation A is also ancestral to C, represented by X variable. This relation is ensured by the first inequality here. Furthermore, we need to ensure that if mutation A and B are both ancestral to C, then either A is ancestral to B or B is ancestral to A. This keeps all ancestral mutation on a single lineage path. Accordingly, this second inequality here ensures that if both XAC and XBC are 1, then at least one of the XAB and XBA is also set to 1. These are all the constraints we used. After getting to know these constraints, let's take a look at the objective functions. As a reminder, we want to minimize the total distance from output or all input trees. We thus find a way to penalize any difference between them. First, if the output tree has B being ancestral to D given by XBD equals 1, but in the input tree T2, B is not ancestral to D, then a penalization of the weight of T2 will be added to the objective. Conversely, if the output tree does not have C ancestral to D, but in the input tree C is ancestral to D, then the objective function helps to add a penalization of also the weight to the objective. Adding up all the penalization, our objective function is shown in this form. To sum up, we have one objective function to minimize the weighted distance from output to all the input, and we used variables X and R to encode the tree, and we have a set of constraints to make sure our output is valid. Next, I want to move on to the results on real data set. We applied our method to a triple-negative breast cancer dataset. First, we used equal weights to all input trees to get to the result tree here. We compared with previous methods, country DP and graphic. We agree with the more recent non-distant-based method, country DP, while previous method, graphic, gave us a slightly different tree. The yellow note highlights the difference where they put MAP3K4 mutations. Our method put MAP3K4 to the top, while graphic put it further down the tree due to the influence of one of the input trees, SciFit. The other two input trees all have MAP3K4 to the top, where the position is more reasonable. Graphic get this tree because it only considers one clustering method to determine how to group mutations into different nodes. We can see how our method overcome some of the limitation of graphic, which got the tree due to restricted clusterings. Now you might wonder, what about weights? What will happen if we put different weights to each of these input trees? We did an experiment to shift weights attached to these three inputs. The ternary plot shows how Tulip worked with different weights for these three input trees. This corner is a weight of 1 on physics, while this corner is a weight of 1 on SciFit, and the weights are sliding in the triangle. The color is the output represent the tree or the set of trees returned. The star is where we put equal weights of all the three trees. The graph is labeled with a number if a set of trees that are tied are returned. Here are 16 ties if we set weight according to this position and scale. As expected, each three input trees dominate the corners, while SciFit tree, which is also the tree returned when we set equal weights, if you remember last slide, physics tree dominate a much larger area with only subtle changes as you vary from these equal weight positions. Is it always like that? The weight doesn't influence the output that much? We want to see how our method work with other data set as well. So we made two similar plots for the ALL patient and also a different triple negative breast cancer patient. We can see these two give us two very different landscapes. On the left, shifting the weights in the center has a more consistent consensus tree outputted as long as we give at least certain amount of minimum support to each input tree. On the right, things can change a lot. Even if we shift a little weight among input, our method TULIP gave us a slightly different output, and some of them are tied. Overall, this experiment shows that our method TULIP helps give a better picture of what the landscape of consensus trees actually looks like when you shift weights, because our method allows allocation of confidence weights to different input trees. That's basically our research. Thank you for listening. I want to thank Leila, my supervisor, for helping me a lot with our research. And of course, this project's done together with Matthew, my research partner. I also want to thank members in Asper Lab. They've all been a great help. Thank you for listening.